Okay, so we'll talk about trace elements. This will be a short discussion lang of the important trace elements. So first is copper. So regarding sources of copper, you have shellfish, liver, nuts, and legumes. Then the absorption of copper is modulated by need, resulting in 55% to 75% rate of absorption. And regarding copper, copper is a component of enzymes or proteins involved in redox reactions. So regarding copper deficiency, cause of copper deficiency first is the increase in intake of zinc because zinc competes with copper in competes with copper in terms of absorption. So the absorption of copper and zinc na competition. So if there's increase in zinc, you have decreased absorption of copper. The next is the association of ceruloplasmin and microcytic and hypochromic anemia. Okay, so it's more of IDA, diba? IDA is microcytic hypochromic anemia. So in ceruloplasmin, ceruloplasmin is a protein that carries or contains copper. Okay, so in order to form ceruloplasmin, you need copper. Okay, so decrease intake of copper, decrease absorption of copper will lead to decreased production of ceruloplasmin. Now, regarding ceruloplasmin, ceruloplasmin is important in the conversion of Fe3 to Fe2. Okay? So, di ba ang ginagamit na to sa atong mga cells, formation of your red blood cell, it's your Fe2. Okay? So, in in conditions with low ceruloplasmin, you have decreased Fe2 concentration. Okay, because ceruloplasmin is important in the conversion of Fe3 to Fe2. Okay, so um, regarding confirmation of the disease aceruloplasminemia, meaning of aceruloplasminemia is the absence of ceruloplasmin, you can administer ceruloplasmin. Okay, and once you introduce ceruloplasmin, there is an increase in iron concentration. Okay? Increase Fe2. Okay? Kaya I think Fe3, di ba, ang siyang ginastore, but ang gina-use is Fe2. Okay? So, in the administration of ceruloplasmin, the effect, you have increase in iron concentration. So, this is an indication nga, so for ceruloplasmin, before the administration of ceruloplasmin, you have decreased iron concentration. But after the administration of ceruloplasmin, you have increase in iron concentration. So that's the confirmatory for aceruloplasminemia. Okay. Next is decreased SOD. Okay, or superoxide dismutase. So superoxide dismutase, the cofactor of superoxide dismutase is copper. Okay, and superoxide dismutase is an antioxidant. So if there's a decreased activity in SOD or superoxide dismutase, you have shortened lifespan of erythrocyte. Okay, so ang erythrocyte early siya mag-hemolysis or dali siya ma-oxidize. Okay, early damage or destruction of red blood cells. Okay, next is next disease-associated copper deficiency have Menkes syndrome. So in Menkes syndrome, we have uh, normal absorption of copper, but there's abnormal transportation of copper. So after absorption, you need to transport the copper from the enterocyte to your plasma. So the problem in Menkes syndrome, you have the problem in the transportation of copper. So, sir, why man I problem with the transportation of copper? Because in Menkes syndrome, we have mutation in the ATP7A gene. Okay, so ATP7A gene codes for the production of CMT1. CMT1 is copper membrane transporter 1. So CMT1, this CMT1 is important in the transportation of um, copper from the enterocyte to your plasma. Okay, so in Menkes syndrome, you have mutation in ATP7A. So the effect, you have decreased CMT1 production. And CMT1, decreased CMT1 production, so decreased transportation of or decreased movement of copper from the enterocyte to your plasma. So the concentration of copper in plasma is low in Menkes syndrome because of the problem with the transportation of copper from enterocyte to plasma. Okay, so that's for Menkes syndrome. Next is copper excess. So for copper excess, cause milk boiled in brass containers. So you have the removal of copper. Next is the disease associated with copper excess. You have Wilson's disease. Okay. So in Wilson's disease, you have overload of copper. Okay. And there's a problem with the transportation of copper out of the liver into the bile. Okay, so the way to remove the excess copper from our system, it's via bile. Okay, now the problem in Wilson's disease, the body cannot transport out the excess um, copper into the bile. Why? Because of the mutation in ATP7B gene. Okay, so for Menkes, it's ATP7A. For Wilson's, it's ATP7B. So this gene codes for the production of ceruloplasmin. Okay, so in Wilson's disease, you have decrease in ceruloplasmin due to the mutation of ATP7B gene. So decrease ceruloplasmin because you need copper to form ceruloplasmin. Okay, no. And that's for Menkes. There's a problem with the formation of the protein, which is ceruloplasmin. Okay, now a problem because of the mutation in ATP7B gene. In Menkes, you have problem with the formation of ceruloplasmin because of copper deficiency. Okay, but in Wilson's disease, you have copper excess due to the inability of the body to produce ceruloplasmin. Okay? So, that's the difference sa imong Menkes and Wilson's. So, it's ATP7B gene that codes for the production of ceruloplasmin. Okay? So, no ceruloplasmin, no excretion of the excess copper into the bile. Okay? So, meaning, the function of ceruloplasmin, diba, is to transport or to excrete the excess copper. The CMT1, di ba, ang CMT1 is to transport copper from enterocyte to plasma. This one, to remove ceruloplasmin, to remove excess copper from your plasma to your bile. Okay? So the problem is the excretion of excess copper in Wilson's disease. So you have deposition of copper in the liver, and other organs. Pwede po siya mo deposit in your brain. Okay? And there's the presence of Kayser-Fleischer ring. So Kayser-Fleischer ring is the deposition of copper sa eyes. Okay? Then, the common remedy, okay, for Wilson's disease is the administration of zinc because zinc competes with copper absorption. Okay? So increase zinc, decrease copper absorption. So treatment, uh, dimecarpol, penicillamine, and ammonium tetrathiomolybdate. These substances chelate copper and increase urinary excretion of copper. Okay, so remember, Wilson's, it's ATP7B gene mutation. Menkes, it's ATP7A gene mutation. Copper excess, it is Wilson. Decreased copper, it is Menkes. Decreased ceruloplasmin, both. Okay. So in Menkes, you have decreased ceruloplasmin due to the decrease in copper. In Wilson's, you have decreased ceruloplasmin due to mutation in ATP7B gene. Okay. Okay, so next is zinc. Zinc, sources of zinc, you have meat and fish. So absorption is inhibited by increased dietary content of phytate or phytic acid. So phytic acid, main source of phytic acid, it is plants, especially kanang seeds and kanang sa mga grains gani. So example, you have oats. Di ba ang oats, it's with covering. That cover or outer layer of oats contains uh, phytate and or phytic acid, and phytic acid inhibits the absorption of zinc. That's why if you consume a lot of grains, it can also cause problem with zinc. Okay, so you have decreased absorption of zinc. Okay, so kanang oats, overconsumption of oats, that can cause decreased absorption of zinc because ang oats contain a lot of phytic acid. Okay, so regarding phytate, phytic acid, that's the principal storage form of phosphorus. Okay, that's phytic acid. And next, it induces synthesis of metallothionein. So metallothionein is produced or synthesized, and one of the components of metallothionein is zinc. Okay. Next, it is transported in the circulation by albumin and alpha-2 macroglobulin. Then the route of excretion or route of excretion, it's gastrointestinal. Then functions of zinc, it's cofactor for more than 3,000 enzymes. So if you have zinc deficiency, you'll have a lot of problems. Okay, there are 300 enzymes, more than 300 enzymes that require zinc as cofactor or activator. Okay, so example of zinc-containing enzymes, you have alkaline phosphatase, alcohol dehydrogenase, carbonic anhydrase, DNA polymerase, and superoxide dismutase. And these enzymes, zinc enzymes, are essential in growth, wound healing, reproductive function, immune system, and protection from free radicals. So if you have zinc deficiency, you'll have problem with growth, wound healing, reproductive function, immune system, and protection from free radicals. Okay, that's why during COVID, diba, during the pandemic, diba, ang singin nila is to take vitamin C with zinc or plus or a multivitamin with zinc. Okay, because zinc is important in the immune system and protection from free radicals. Okay, and remember, regarding zinc, I think na-mention man ako na siya sa discussion regarding vitamins. In order to produce the active form of vitamin A, you need zinc as cofactor. Okay? The next is for zinc deficiency causes. You have malabsorption, chronic disease of the liver, and alcoholism, and diet high in phosphate or diet high in phytate or phytic acid, or diet low in zinc. Okay, so for laboratory evaluation of zinc, you can measure metallothionein because metallothionein, one of the components in metallothionein is zinc. So decrease zinc, you have decreased metallothionein concentration. Then next is you have cobalt. So for cobalt, it's the constituent of B12. That's why the other name of B12 it's cobalamin because it contains cobalt. Okay. The next is chromium. So chromium, it's important for type 2 DM. So chromium supplementation has demonstrated improved glucose tolerance, meaning for those individuals with type 2 DM or insulin resistance, uh, chromium is helpful in bringing back the sensitivity of the receptor to insulin. Okay. Next is selenium. So selenium is incorporated into proteins, notably glutathione peroxidase. Okay. So regarding glutathione peroxidase, this one is an antioxidant. Okay. So one of the components of your glutathione peroxidase is selenium. Okay. So decrease selenium, you have decreased glutathione peroxidase, which is important in converting free radicals to detoxified radicals. Okay. So selenium supplementation, there are studies, the result about selenium supplementation is it decreases risk of cancer. Okay, so if you take selenium, you have decreased risk of developing cancer because Selenium is a component of your glutathione peroxidase, and glutathione peroxidase is an anticoagulant. Okay?